All right, let's dive into a masterclass on pharmacodynamics. This is all about what a drug actually does to your body. We're going to break down exactly how these chemicals work, from the very first moment they latch onto a cell, all the way to the final effect you feel. So, let's get started.
Here's our roadmap for today. We'll kick things off with the fundamental principles, then get into how we actually measure a drug's effect. After that, we'll take a deep dive into receptors, the drug's main target, and explore the different ways drugs can act. Then we'll see how a drug's effect can change over time, and wrap up by looking at all the different factors that can influence how a person responds to medication.
Okay, first up, the fundamentals. Before we can get into the nitty-gritty, we have to make sure we're all on the same page with the core concepts that define this whole field. So, what is pharmacology? Simply put, it's the study of how drugs and our bodies interact. And what's a drug? Well, it's any chemical we introduce from the outside, that's what exogenously means, that changes how our body normally functions. And here's a really important note right off the bat. Most drugs out there aren't cures. They're designed to manage and treat symptoms. Now, pharmacology is split into two big areas. There's pharmacokinetics, or PK, which is all about what the body does to the drug. You know, how it absorbs it, moves it around, breaks it down, and gets rid of it. But that's a topic for another day. Our focus today is pharmacodynamics, or PD. This is the flip side, what the drug does to the body. This covers everything from its therapeutic benefits to its toxic side effects, where it acts, and exactly how it works, which, a lot of the time, involves either copying or blocking the natural molecules our body already makes.
Okay, on to our second section. We know drugs produce an effect, but how do we actually measure that? How do we put a number on it? This is all about taking that initial drug interaction and turning it into something we can see and quantify.
The whole process really follows a simple cause and effect pathway. Step one is the interaction. The drug finds and binds to its target molecule, which we call a receptor. Step two is signal transduction. That binding event kicks off a chain reaction, a whole cascade of biochemical signals inside the cell. And step three is the physiological response. All those signals lead to a real, measurable outcome, like your blood pressure dropping or a headache going away. To see this relationship between dose and response, we use graphs. Now, if you just plot the data directly, you get this hyperbolic curve on the left. It's okay, but not super useful. So what we do is we use a semi-log scale for the dose, and that transforms the graph into this much more useful S-shaped or sigmoidal curve on the right. The reason this shape is so much better is that steep, almost straight line in the middle. That's the drug's sweet spot, its therapeutic range, where a small change in dose gives you a big, predictable change in the effect. Now, this is where these curves get really powerful because they let us compare different drugs. Look at this graph with drugs A, B, and C. See how their curves are in different places? One is shifted to the left and one is taller than the others. These visual clues are telling us about two absolutely essential properties of any drug, its potency and its efficacy. So let's lock down those definitions. Potency is all about the amount of drug you need. We measure it with a value called EC50. That's the concentration needed to get 50% of the maximum effect. And here's the key. A lower EC50 means a more potent drug, because you need less of it to get that half maximal effect. Efficacy, on the other hand, isn't about the dose. It's about the maximum possible effect the drug can produce. We call this Emax. It's the absolute ceiling for that drug, what you get when every single receptor is occupied. So just think, potency is how much, efficacy is how well. And this leads us to an incredibly important clinical rule of thumb. A drug with greater efficacy is almost always better than one that's just more potent. Why? Because efficacy tells you the absolute best the drug can do. I mean, who cares if a drug works at a tiny dose if its maximum effect is super weak? What really matters for a patient is getting a strong, meaningful response. And that, my friends, is all about efficacy.
Okay, so far we've been talking about the graded response in just one person. But to figure out if a drug is actually safe for widespread use, we have to look at how it affects a whole population. And for that, we switch to a different tool, the quantile dose-response curve. This isn't about the size of the effect anymore. It's a simple yes or no, all or none question. What percentage of the population showed a specific response at this dose? This type of curve gives us some incredibly important numbers. There's the ED50, or median effective dose, the dose where half the population gets the therapeutic benefit. Then there's the TD50, the median toxic dose, where half the population experiences side effects. And finally, the LD50, the median lethal dose. By comparing the toxic dose to the effective dose, literally just dividing TD50 by 8050, we get the therapeutic index. This number is a crucial measure of a drug's safety. A big index means there's a wide gap between a helpful dose and a harmful one, which makes the drug much, much safer.
All right, Section 3. Let's talk about receptors. We've mentioned them a bunch already, but now we're putting them center stage. Think of these as the molecular gatekeepers, the specific targets that drugs are designed to find and interact with. They are truly at the heart of how drugs work. So most drugs target one of 4 major families of proteins. And what's really fascinating is that their structure completely dictates how fast they work. Check out the timeline at the bottom. The ligand-gated ion channels, they respond in milliseconds. Boom. G-protein-coupled receptors take a few seconds. But the kinase links and nuclear receptors, they're the slow burners, taking hours to work because their job often involves going deep into the cell and actually changing gene expression. Let's run through them. First, ligands-gated ion channels. These are basically molecular pores. A great example is the nicotinic receptor, where succinylcholine acts as an agonist to open it, while tubulurine is an antagonist that blocks it. Second, G-protein-coupled receptors, or GPCRs. The asthma drug salbutamol hits the beta-2 adrenergic receptor, firing up G-proteins and second messengers to relax the airways. Third, kinase-linked receptors. These phosphorylate proteins. The cancer drug trastuzumab targets the HER2 receptor, while peginterferon alpha-2A uses a similar mechanism. Fourth, we have nuclear receptors, which are found inside the cell. Glucocorticoids bind to these to change which genes are being transcribed. And finally, some drugs skip receptors altogether and target intracellular enzymes directly. The most famous example is aspirin, which works by inhibiting the COX enzyme.
Let's zoom in on that GPCR pathway because it's so common and just elegant. When the drug hits the receptor, it's like the first domino in a chain. It activates a G protein, which then turns on an enzyme. That enzyme starts cranking out tons of tiny molecules called second messengers, like CAMP and IP3. These little messengers then spread throughout the cell, activating other enzymes called protein kinases, which leads to a huge amplified response, all from that one single drug molecule binding.
So what actually makes a drug stick to its receptor? It all comes down to chemical bonds. You've got covalent bonds, which are like superglue, incredibly strong and often permanent. Then there are ionic bonds, which are like the attraction between 2 magnets. Hydrogen and hydrophobic bonds are crucial for getting that perfect specific fit. And finally, you have van der Waals forces. They're the weakest individually, but when you get a bunch of them together, they really add up. The specific combination of these forces determines how well a drug binds. Okay, on to Section 4. So, a drug binds to a receptor. What happens next? What does it actually do? This is where we get into the different profiles of drug action. It all boils down to 2 words we've already learned: affinity and efficacy. Affinity is just the ability to bind. Efficacy is the ability to activate the receptor after binding. An agonist has both. It binds and it activates a response. An antagonist, though, has affinity but 0 efficacy. It's like a key that fits perfectly into the lock but then snaps off. It's stuck in there, so it's not activating anything, and now the real key can't get in to do its job. This graph just lays it all out beautifully. You can see the full agonist way up top, causing a big response. The partial agonist is trying, but it just can't reach that same height. The antagonist is just sitting there at the baseline, doing nothing on its own. And then you've got the inverse agonist, which is actually pulling the activity below the baseline. Let's define these properly. A full agonist is simple. It gives you the 100% maximum response. A partial agonist is a bit strange. It activates the receptor, but even if it binds to every single one, it can't produce that full effect. And here's the crazy part. In the presence of a full agonist, it actually acts like an antagonist, getting in the way. An inverse agonist works on receptors that are naturally a little bit on all the time. It binds and forces them to shut down completely. Finally, a biased agonist is a very sophisticated drug that tells the receptor to use one signaling pathway but not another, which is a clever way to reduce side effects. All right, Section 5. Now, a drug's effect isn't set in stone. It can be blocked by another drug, or its own effect can just sort of fade over time. Let's talk about how and why that happens. Okay, this is a classic exam topic: competitive versus non-competitive antagonists. A competitive antagonist fights for the exact same binding spot as the agonist.
It's a head-to-head battle. You can overcome it by just flooding the system with more agonist. That's why the Emax, the maximum effect, stays the same, but the curve shifts to the right. You've reduced its potency. Now, a non-competitive antagonist is different. It binds to another spot on the receptor, changing its shape, or it just binds irreversibly. No amount of extra agonist can overcome it. So you don't change the potency, but you crush the efficacy. The maximum effect goes down.
You've probably heard of drug tolerance, when a drug just doesn't work as well over time. If it happens really fast, we call it desensitization or tachyphylaxis. The slower, more gradual process is what we call tolerance. And the body has a whole toolkit to make this happen. It can change the receptor itself, or pull the receptors inside the cell so the drug can't find them. It can run out of the signaling molecules the drug needs. The body can even get better at metabolizing the drug, or it can create an opposite physiological effect to cancel it out. Some cells can even just actively pump the drug right back out. And that brings us to our final section. This is where the textbook hits the real world. We've gone over all these beautiful, clean principles, but in reality, things get complicated. The way any one person responds to a drug is affected by a ton of different variables. We can sort these factors into two buckets. First, there are patient factors, things like body weight, age, and any other diseases a person might have. But the huge one here is genetics. A tiny difference in someone's DNA can completely change how they handle a drug, making a normal dose either toxic or totally useless. Then you have drug-related factors. Was the drug administered correctly? Is the patient actually taking their medicine? Are they on other drugs that might interfere? And, of course, have they developed a tolerance? So we end on this big, central question. We have all this incredible science, but the real challenge is applying it to the individual person. With so many variables, from genetics to lifestyle, all affecting the outcome, how do we get from these general principles to a safe, effective treatment that's perfectly tailored for every single patient? That question right there is the driving force behind the entire field of personalized medicine.